Cutaneous vasculature. Overview of the skin's vasculature. Main divisions. The cutaneous vasculature is organized into three interconnected networks: superficial vascular plexus, deep vascular plexus, and subcutaneous vascular plexus. Microvessels are the fundamental units of the system, consisting of endothelial cells, a single cell layer that lines the vessels, and mural cells, which support cells, including pericytes, PCs, and smooth muscle cells, SMCs. The microvessels have cellular diversity, which host various resident perivascular leukocytes, T cells, macrophages, etc., which act as sentinels for infection and injury. Key functions of skin microvessels have special roles in regulating blood fluidity, forming a barrier, controlling local blood flow, and thermoregulation, unique and critical. Structure and organization. Superficial vascular plexus. It is a network of paired arterioles and venules located just below the epidermis. Capillary loops arise from the arterioles, loop upward into the dermal papillae, and then return to the venules. Deep vascular plexus is a deeper network of anastomosing vessels parallel to the superficial vascular plexus. It connects to superficial vascular plexus via short, straight vessels. It provides nourishment for adnexal structures such as hair follicles and glands. It also serves as a portal for leukocytes to enter the dermis during inflammation. Dermal veins form a complex, polygon-like anastomosing network. This is the architecture of skin vasculature. Structure of vessel walls. Arterioles. Wall has three layers. Intima is lined with endothelial cells. Media consists of smooth muscle cells in larger arterioles, or pericytes in the smaller terminal arterioles. Adventitia is outer connective tissue layer. Veins and venules. Structurally similar to arterioles but with larger lumen and thinner muscular walls. Also contain valves to prevent backflow of blood. Capillaries and postcapillary venules. Capillaries are narrow lumen, lined by a single file of tightly connected endothelial cells. Postcapillary venules are wider, looser endothelial junctions and a thicker, multilayered basement membrane. This is the schematic structure of capillaries. Properties of and functions. Hemostasis, perm selectivity, perfusion, thermoregulation, and immune surveillance. Role of endothelial cells in hemostasis. Endothelial cells are the main regulators of keeping blood fluid. They do this through anticoagulant and antiplatelet functions. Anticoagulant functions. Even under normal, basal conditions, endothelial cells express inhibitors to prevent unwanted clotting. Key examples. Thrombomodulin changes thrombin's role so it activates protein C, which then shuts down clotting factors. Heparin sulfates help activate antithrombin 3, inhibits thrombin and factor Xa. Tissue factor pathway inhibitor blocks the activation of the clotting cascade early on. Antiplatelet functions. ECs also prevent platelet activation. They release nitric oxide NO and prostacyclin PGI2. Both stop platelets from sticking or activating. They also use ectoenzymes to break down activating signals like ADP and ADP. Anticoagulant function. Heparin sulfates activate thrombin 3, block factor Xa and thrombin. Thrombomodulin activate protein C, protein S, inactivate factor 5 and 8. TFPI blocks tissue factor 7A complex. Antiplatelet functions. ECs also stop platelets from activating and sticking. Ectoenzymes convert ADP and ADP into adenosine monophosphate, which cannot activate platelets. Nitric oxide and prostacyclin both are strong inhibitors of platelet activation and aggregation. ECs form a barrier that prevents platelets from contacting basement membrane collagen, a strong platelet activator. Perm selectivity and perfusion. Perm selectivity, the ability of the EC lining to act as a barrier. It allows limited passage of water and small solutes but restricts the movement of macromolecules and cells. Barrier structures. Tight junctions. In arterioles and capillaries, they prevent the paracellular passage of macromolecules. Adherens junctions. In post-capillary venules, these are looser and can be dynamically regulated to allow macromolecules to pass, especially during inflammation. Perfusion, the control of blood flow through the microvessels. This is crucial for local tissue nourishment and in the skin for thermoregulation. This diagram shows how endothelial barriers regulate the passage of water and solutes across microvessels. Starting in the arteriole, the wall has smooth muscle cells for tone and tight junctions that restrict passage of large macromolecules. Hydrostatic pressure pushes water outward into tissues. In the capillary region, there is a shared basement membrane and parasites that support the endothelium. Red blood cells remain confined, while small solutes and water can move depending on pressure gradients. Moving to the post-capillary venule, endothelial cells are connected by adherens junctions, which are more permeable. They allow small molecules and some fluid to pass. Here, oncotic pressure from plasma proteins draws water back in. Control of perfusion by regulating smooth muscle cell tone. The active resistance of blood vessels can be modulated by altering the contractile tone mural vascular smooth muscle cells in muscular arteries and arterioles. Endothelial cells can produce vasodilators such as nitric oxide and prostacyclin. NO directly activates soluble guanylate cyclase to elevate cyclic guanosine monophosphate, cyclic GMP levels, and PGI2 interacts with a membrane G-protein coupled receptor that activated adenylate cyclase to increase cycloadenosine monophosphate, CAMP. Both reduce the degree of actomyosin contraction. Endothelial cells can also produce vasoconstrictors such as endothelin, ET, or, via the ectoenzyme angiotensin-converting enzyme, ACE, convert inactive angiotensin 1 at 1 to vasoactive angiotensin 2 at 2. Both ET and ATII signal through G-protein-coupled receptors to increase the level of cytosolic free calcium ion, increasing the degree of actomyosin contraction. Nerves may release neurotransmitters that also bind to G-protein-coupled receptors to increase cytosolic free calcium and actomyosin contraction. Increased actomyosin contraction shortens the length of the smooth muscle cells, which are arranged radially, thereby reducing the vessel lumen diameter and increasing resistance to flow. Thermoregulation. Unique role. The control of blood flow in skin microvessels has a special and critical role in thermoregulation that isn't seen in other parts of the vasculature. Mechanism. In hot conditions, blood vessels in the skin dilate vasodilation to increase blood flow to the surface, allowing heat to dissipate from the body. In cold conditions, blood vessels constrict vasoconstriction to reduce blood flow to the skin, conserving heat for the core organs. This process is a major part of the body's overall thermal regulation system. Thermoregulation in non-glabrous and glabrous skin. Non-glabrous skin uses a multimediator system for fine control, while glabrous skin uses a simple on or off switch that is opening and closing of arteriovenous shunts, together providing a highly efficient thermoregulatory system. Immune surveillance. The skin is both a physical barrier and an immunologic barrier. Resident immune cells. Several immune cell types are always present. Keratinocytes produce cytokines and antimicrobial peptides. Endothelial cells, immune regulators, express MHC. Macrophages, Langerhans cells, and dermal dendritic cells. Resident T cells and recirculating T cells. Skin-associated lymphoid tissue, SALT. This system coordinates immune surveillance. Includes memory T cells, cutaneous lymphocyte antigen (CLA), chemokines, and circulating dendritic cells and B cells. Endothelial role. Endothelial cells in skin can express MHC class I and II, along with costimulatory molecules. This allows them to present antigen and activate T cells. T cell response. Once an antigen is recognized, T cells undergo diapedesis, migration through endothelium into the skin. This provides local, targeted immune defense. This shows how specialized T lymphocytes are guided from the blood into the skin. These T cells carry the receptors CCR4 and CCR10. First step. Venules in the dermis release TARC, CCL17, which binds CCR4 and pulls the T cells out of circulation. Next step. Once in the dermis, the cells follow a gradient of CTAK, CCL27, produced by the epidermis. This binds CCR10 and directs the cells upward into the epidermis. The result is targeted homing of T cells into the skin for immune surveillance. Important point is, while this system protects against pathogens, if it becomes overactive, it contributes to inflammatory skin diseases like eczema.